Welcome to the fundamentals of biomolecules. Today, we are going to explore the fascinating world of carbohydrates, specifically focusing on monosaccharides and disaccharides. Think of this as learning about the fundamental building blocks of life itself. By the end of this session, you will understand exactly how your body stores and uses energy at a molecular level. Let us dive in and discover the chemistry that keeps us alive. To begin our journey, imagine you are playing with a giant box of Lego bricks. Every massive castle you build is made up of individual, unbreakable single bricks. In biology, carbohydrates are like those massive Lego structures, and monosaccharides are the single, unbreakable bricks. The word comes from Greek, meaning one sugar. When you eat fruit or bread, your body breaks down the complex food into these tiny single sugar molecules. They are the simplest form of a carbohydrate, meaning they cannot be broken down or hydrolyzed into smaller units. This makes them the absolute fundamental starting point for understanding biological energy. Now that we know what these single bricks are, let us look at how they are built and what they do. Chemically, monosaccharides are polyhydroxyaldehydes or ketones. This simply means they have a carbon chain with one double-bonded oxygen and multiple hydroxyl groups. If that double bond is at the end, it is an aldose. If it is tucked inside, it is a ketose. Why do we care? Because these tiny molecules, like glucose, are the primary fuel source for almost all living cells, especially your brain. Furthermore, they are vital structural building blocks. For instance, the sugar ribose forms the structural backbone of your DNA and RNA. Understanding their biological role naturally leads us to their chemical makeup. This brings us to the general empirical formula for a basic monosaccharide. The formula is CnH2nOn. Conceptually, this tells us that for every one carbon atom, there are exactly two hydrogen atoms and one oxygen atom. This is exactly why they are called carbohydrates, meaning carbon plus water. In this formula, C represents carbon, H represents hydrogen, and O represents oxygen. The variable N represents the total number of carbon atoms in the sugar molecule, which typically ranges from 3 to 7. If the value of N increases, the molecule becomes larger and heavier, fundamentally changing its physical properties. Before we move on, let us see this formula in action with a concrete example. Let us determine the exact chemical formula for glucose, which is classified as a hexose sugar. We know glucose is the primary fuel for our bodies, so finding its exact atomic composition is crucial. The prefix hex means 6, so we know immediately that a hexose sugar contains exactly 6 carbon atoms. We set our variable N equal to 6. By substituting 6 into our general equation, we multiply the hydrogen count by 2, giving us 12 hydrogens. The oxygen count remains 6. This gives us the final chemical formula for glucose, which is C6H12O6. Having calculated the formula for an energy sugar, let us apply the same logic to a structural sugar. We will find the chemical formula for ribose, a pentose sugar that forms the backbone of genetic molecules like RNA. The prefix pent means 5, indicating a backbone of exactly 5 carbon atoms. We set the variable N to 5. Substituting this into our empirical formula, we multiply the hydrogen count by 2, yielding 10 hydrogens, while the oxygen count matches the carbon at 5. The final chemical formula is C5H10O5. This physically represents the exact molecular structure your body synthesizes for RNA, proving how changing just one carbon atom completely alters the biological function. Now that we have calculated their formulas, let us visualize what these molecules actually look like. On the screen, you are seeing the chemical structures of two hexose sugars, glucose on the left and fructose on the right. Notice that both have exactly 6 carbon atoms, 12 hydrogens, and 6 oxygens. However, look closely at the highlighted carbonyl groups. In glucose, the double-bonded oxygen is at the very top of the chain, making it an aldohexose. In fructose, the double-bonded oxygen is on the second carbon, tucked inside the chain, making it a ketohexose. This diagram perfectly illustrates how molecules with the exact same chemical formula can have completely different structural arrangements, known as structural isomers. Seeing how single sugars are structured brings us to an important question. What happens when we want to build something bigger? Just like snapping two Lego bricks together, when two monosaccharides join hands, they form a disaccharide. This literally translates to two sugars. To get a good grip, they have to drop a few atoms to make room for the connection. This covalent linkage is specifically called an O-glycosidic bond. It is formed through a dehydration synthesis, also known as a condensation reaction. This means that as the two sugars join, one molecule of water is physically removed from them. This connection is the secret to how plants transport sugar and mammals provide energy-rich milk. Now that we understand how they connect, let us explore why these disaccharides are so incredibly important for living organisms. They are essential for the safe transport and efficient storage of energy. For instance, plants produce glucose in their leaves, but transport it to their roots as sucrose, or table sugar. Sucrose is less reactive, so it will not accidentally react with other molecules along the way. In humans, lactose is the primary disaccharide found in breast milk, providing a dense, easily digestible energy source for infants. However, to use this energy later, we need specific enzymes to break these glycosidic bonds. Without them, we suffer from digestive issues like lactose intolerance. This biological bonding process can be perfectly described using a mathematical equation. Here is the formula representing the condensation reaction that forms a disaccharide. Conceptually, it shows two separate sugar molecules coming together, losing water, and forming a new, larger molecule. The first term represents the atomic composition of the first monosaccharide. The second term represents the second monosaccharide. The minus H2O signifies the crucial removal of one water molecule, consisting of 2 hydrogens and 1 oxygen during the bonding process. Finally, the arrow indicates the chemical transformation into the final disaccharide product on the right side. If more sugars join, more water molecules are subtracted for every new bond formed. Let us put this condensation formula to work with a real biological example. We will calculate the exact chemical formula for maltose, a disaccharide formed by joining 2 molecules of glucose together. We are given 2 glucose molecules, each with the formula C6H12O6. First, we mathematically add the atoms of both glucose molecules, giving us an intermediate count of 12 carbons, 24 hydrogens, and 12 oxygens. But we cannot stop there. Because a glycosidic bond is formed, we must subtract 1 molecule of water, which is 2 hydrogens and 1 oxygen. This leaves us with the final, stable chemical formula for maltose, which is C12H22O11. Having successfully calculated maltose, let us try one more highly practical example: the formation of sucrose, or common table sugar. Sucrose is formed by joining one molecule of glucose and one molecule of fructose. We know both glucose and fructose are hexose sugars, sharing the exact same chemical formula of C6H12O6. Therefore, our mathematical approach is identical to maltose. We add the formulas together to get our initial total of 12 carbons, 24 hydrogens, and 12 oxygens. Then, we subtract the H2O molecule required to form the O-glycosidic bond. The final formula is C12H22O11. This proves that maltose and sucrose share the exact same formula, despite behaving completely differently in our bodies. To truly grasp this dehydration synthesis, let us look at a visual representation of the reaction we just calculated. On the screen, you see two individual glucose molecules on the left. Notice the highlighted hydroxyl groups on the adjacent carbons. As the reaction proceeds to the right, indicated by the arrow labeled dehydration synthesis, those specific atoms are removed to form a single water molecule, shown as minus H2O. The two glucose molecules are now covalently linked by a single oxygen atom, forming the O-glycosidic bond. The resulting massive molecule on the right is maltose. This diagram beautifully illustrates how nature builds complex, stable structures from simple, reactive building blocks by simply removing water. We have covered a tremendous amount of ground today, so let us summarize the most important ideas. First, monosaccharides are the fundamental, indivisible units of carbohydrates, serving as primary energy sources and structural backbones. Second, they strictly follow the empirical formula, where carbon and water are in a 1-to-1 ratio. Finally, disaccharides are formed when two monosaccharides link together via an O-glycosidic bond, releasing a molecule of water in a process called dehydration synthesis. You now understand exactly how simple sugars combine to form the vital transport and energy storage molecules that power all living systems. Excellent work mastering these fundamental biomolecules.